This next series of slides are kind of a catch-all for a whole bunch of seemingly random processes that can be targeted to inhibit immunology in a lot of different contexts. So the first target is going to be inhibition of co-stimulation. Okay, and so kind of the prototypical drug is abatacept, and we're looking at the ability to kind of shut down that co-stimulatory B7 molecule on the surface of antigen-presenting cells. And so remember, this antigen-presenting cell is going to interact with a T cell, and then that complex is going to actually induce a change in some of the proteins called co-stimulation, and that's going to increase the activity of some of these T cells. What this abatacept does is it actually blocks co-stimulation, and so a lot of the T cells are really not going to be able to properly have their effector function because the co-stimulatory ligand has been severely restricted. And that's important because the reason that it was restricted was because it was basically inhibiting that antigen-presenting cell interaction. And so the antigen-presenting cell was actually going to be directing the T cell as to which cells it should go after, and now it can't do that. So clinically, we'll use this drug for rheumatoid arthritis. And so again, these would be diseases or indications that are being driven by T cells that are targeting autoreactive antigens. And so in RA, we'd be looking at antigens that are being processed incorrectly from something like the synovium, and you see this really potent directed T cell response. This abatacept and others that can inhibit co-stimulation would negate that. So this drug is injected. Adverse reactions are listed there. You can read them by yourself. I'm not sure that STEP is going to go into that great detail in terms of this random immunosuppression type drug, and then what's the possible weird, unique adverse reaction. I don't see that, but it is there in case you would like to know. And this is what that co-stimulation process looks like. And this is kind of very similar in terms of this reaction right here. In the previous slide where we looked at cyclosporine and tacrolimus, obviously the machinery is different, but it involves this reaction between an antigen-presenting cell and a T cell. Okay, and then this co-stimulatory receptor, which is basically CD28. All right. Another mechanistic approach would be to block cell adhesion. We can accomplish that with the monoclonal antibody Bevacizumab, and this is going to block the T cell adhesion molecule ICAM or CD11A. Typically, we'll see these drugs used for moderate to severe plaque psoriasis. There is an increased risk of JC virus kind of re-emerging, leading to PML. And then there's an increased risk for immune-mediated thrombocytopenia. We can inhibit complement through eculizumab, and so this is going to be an antibody directed against C5, which will inhibit or prevent complement activation. Clinically listed there for some rather very specific and somewhat rare clinical indications. Other molecular targets we can utilize would be inhibition of angiogenesis. We can accomplish that using the monoclonal antibody Bevacizumab, which is Avastin. So this is a monoclonal antibody to the VEGF receptor. We're going to shut down angiogenesis, and we can see this used as an adjuvant therapy for a whole host of solid tumors, ovarian, lung, colorectal, brain, etc. And there's multiple dosing regimen guidelines and protocols, and we're not going to get into that. Obviously recognize that there are several VEGF receptors. Bevacizumab would be specific to one or more, and there's multiple drugs out there that can be used to really kind of restrict this VEGF dependent cascade. And again, you don't need to know all this because this is a huge body of literature, and unless you're going to be an oncologist specializing in adjuvant therapy for solid tumors, you don't need to know this. Okay, tons of trials right now looking at uh, some of the more non-VEGF mediated pathways for angiogenesis. So there's multiple ways of kind of shutting this pathway down, and there's a huge body of research and a huge body of people working on this. Okay, um, let's stop there, and then we'll pick up on the next unit shortly.